Hi, everybody. I'm Andy Miller. I'm a professor here at UMBC in the Department of Geography and Environmental Systems, and I host our seminar series. And in just a moment, I'm going to turn the floor over to Rhonda Plofkin, who will introduce this week's speaker. Before we do that, I'm just briefly going to put up on the screen an announcement of next week's seminar for those who might be interested. Our seminar speaker next week is Keith Eshelman, who's a professor at the Appalachian Laboratory, and his topic will be detecting and understanding hydrologic change in developing watersheds. Is green stormwater infrastructure making a difference? So we will be sending out a link to that. Anybody who's not on our regular mailing list who is interested in getting that link is free to send me an email, and I'll make sure we put you on the distribution list for that seminar. I'll put my email address in the chat after we get going. So I'm going to go ahead and turn it over to Rhonda Plofkin, who's one of our graduate students. Rhonda, I'm moving you to the stage so that you can go ahead and introduce our speaker.
Hello, everybody. I am introducing Dr. Holly Michael today. Thank you, Dr. Michael, for being here with us. Dr. Michael is a Unidel Fraser Russell Chair in the Environment and professor in the Department of Earth Sciences and Civil and Environmental Engineering at the University of Delaware. She's also director of the Delaware Environmental Institute. She holds a BS in Civil Engineering from the University of Notre Dame and a PhD in Hydrology from the Massachusetts Institute of Technology. Her research interests include water resource management, contaminant hydrology, coastal hydrogeology, groundwater surface water interactions, and geostatistics. Some of her current projects include investigating groundwater flow into estuaries, modeling groundwater salinization due to climate change, evaluating sustainability of arsenic-safe groundwater in Bangladesh, and application of experimental economics to groundwater resources. So with that, I think Dr. Michael.
Thanks so much, Rhonda, for the nice introduction, and thanks, Andy, for the invitation. I'm really happy to be here and to talk with all of you. So I will share my screen. See? Okay. Can you see the full screen? Not in speaker mode? Good, okay. Great. So I'm going to talk about a couple of different projects. Some of the work is finished, is published, and some of it is really new. So I'll be talking about a new critical zone network project, which is a similar project to the urban CZN that I'm sure some of you are involved in. And so some of the data I'm going to show you is really new, but I want to give you a sense of what we're doing and the instrumentation we have, and think about ways that we could potentially even connect the projects and collaborate. So I want to acknowledge my co-authors. There are many of them, and I'll also acknowledge them throughout the seminar. It's not advancing. There we go. Okay. So this is a schematic of a coastal environment. On the land side, we have freshwater. So you can see there's a cross-section here where we have fresh groundwater flowing toward the sea. And then we have saline groundwater under the ocean, which exchanges with the ocean. And there's an interface between the freshwater and the saltwater, which is sort of angled like this because the freshwater is more buoyant than the saline water. These environments are really important, as we all know. They mediate fluxes from land to sea, carbon, nutrients, contaminants. They are really interesting to study, I think, because they're so complex, hydrologically, biogeochemically, ecologically, and adding in the human component makes it even much more so. And they're also highly dynamic over multiple time scales. So coastal environments are acted on by waves, by tides, by seasons, and by millennial scale changes in sea level. And so I think we need to work across these scales, across different settings and across disciplines to be able to estimate land sea fluxes of various constituents and to predict how they might evolve in the future. So coastal wetlands are no exception to that coastal complexity idea. They are hydrologically complex because you have fresh water coming in from land, both overland and also through the subsurface. And you have saltwater input through these tidal channels and also directly from the ocean or the bay. They're ecologically complex. So if you go out into a salt marsh, you'll see very distinct zonations of vegetation. And biogeochemically, they are hotspots where nutrients are processed and stored. And also, really importantly, they have high rates of carbon burial. But there is complex nutrient cycling and complex carbon dynamics within this system. So both emissions of CO2 and uptake, but also fluxes between the marsh and the tidal channels and the ocean. The marshes have a lot of ecosystem services, right? They're fishery and coastal wildlife habitat. They protect the upland from storm surges. They trap and break down contaminants. And as I mentioned, they sequester carbon. Forests sequester about up to 10 grams of carbon per meter squared per year. Tidal marshes are up to 1,000 times that amount. So per square footage, marshes are really valuable in terms of sequestering carbon and, of course, then mitigating climate change. But the other thing is that they're right on the coast, so they're really vulnerable to sea level rise and other climate and human pressures. Data show that salt marshes are losing about 1% to 2% of their area each year. So that's a huge amount, considering especially that we don't have a huge area of salt marsh compared to tropical forests. It's about an order of magnitude less. So again, understanding all of these things, I think we need to get at how they're all linked in order to be able to predict into the future as things change on the coastal landscapes, how these fluxes between land and sea, how the carbon sequestration are going to change. So that's what we're trying to do. And I'll tell you first about this project that was led primarily by my former graduate student, Julia Guimond, who is soon to be an assistant scientist at Woods Hole Oceanographic Institution, and also collaborators Kevin Moffat and Angelia Seifereth. So in this project, we were looking at the physical, biological, geochemical linkages that impact carbon fluxes and marsh migration. So we worked at the St. John's National Estuarine Research Reserve. This is in Delaware, along the Delaware Bay. Backing up a little bit, this is what it looks like. This is the, can you all see my cursor?
Yes.
Hopefully you can. Okay. This is the St. John's River, and this is a tidal channel that comes through. This is the reserve and a boardwalk that I'll show you later. So the idea is, this is a overly complex schematic of a salt marsh. So here we have the tidal channel, and we have the groundwater table in this blue dashed line, and we can see water levels in these wells that go up and down over time in response to the tides in the tidal channel. And there are variations in topography along the marsh, which determine, one, how influenced they are by the tidal creeks, but also their biogeochemistry. So for example, we often see high EH in areas that aren't inundated very often, where you have a thicker unsaturated zone, and as you go deeper, a lower EH beneath the water table. And of course, so this is redox condition, right? And of course, those redox conditions affect the cycling. And what I'm going to talk about primarily today is carbon, and thinking about the fluxes of carbon. One is the horizontal flux of dissolved inorganic and organic carbon between the marsh platform and the tidal creeks. I'm actually not going to talk about that much today, but if you're interested, just let me know. And primarily I'll be talking about the factors that lead to vertical fluxes, primarily of carbon dioxide. And so the primary reaction that relates to that is the aerobic respiration of organic matter to carbon dioxide. So under oxic conditions or high EH conditions, organic matter is converted to inorganic carbon to carbon dioxide, which then can flux vertically into the atmosphere. So this is our salt marsh. Again, this is the St. John's Reserve. We have a lot of instrumentation. We have wells in the upland. We have level loggers in the channel, shallow monitoring wells. This is a zoom in so you can see that we have transects along the tidal creek and also in different areas of the marsh. And to give you a sense of what we were doing, we made a lot of measurements. So hydrologic measurements, we have monitoring wells that have water pressure, temperature, and salinity or conductivity. We have piezometers and seepage meters that we use to measure the groundwater surface water interactions between the tidal channel and the marsh. This is Julia. She's doing slug tests in the wells to measure the permeability of the marsh sediments. And we also did some biology and geochemistry. Julia did quite a bit of work thinking about how the crabs affect the hydrology, and I don't have time to talk about that today, but these are fiddler crab burrows, and these are casts that she made of those burrows, and she also counted them over time, and actually the crabs have quite a bit of influence on both the spatial and temporal variability and permeability. But that's another side story that I'm happy to talk about later. And we measured organic and inorganic dissolved carbon concentrations, and we have these multi-depth redox sensors. These are platinum electrodes mounted on a fiberglass pole that we can put down into the marsh sediments and measure redox every 15 or measure EH every 15 minutes at different depths. So what Julia found is that the ecological zonations that we see in the vegetation really correspond to hydrological zonations. And so again, here's our outline of our salt marsh. And if we look at the water levels, so these are groundwater levels measured in wells in the different zones. Here I'm showing water table elevation over time. And the black dashed line is the elevation of the land surface. So you can see that there's tidal variation. So each of these ups and downs is a semi-diurnal tide. And during spring tides, you get a substantial amount of inundation, but for the most part, it's inundated twice a day, right? In this zone, the spring neap zone, which is a little bit higher elevation, the diurnal tidal fluctuations are fairly small, but there are pretty big changes on the 2-week spring neap cycle. And so the water levels come up during the spring tides and then are much lower during the neap tides. And so you only have flooding during the spring tides. And then in the interior of the system, the water levels are almost always above land surface. So even though they're varying, it's actually groundwater flooding. It's not flooding from overland. It's just coming up from the subsurface. And it's almost always inundated. And so there are spatial and temporal changes in hydrology that then cause differences in redox conditions. So these are those redox probes that I mentioned. We put them next to our wells in the marsh sediments. And here are some of the results of those. So on the y-axis is the EH, or the redox potential. And on the x-axis is time. So this is about a week.
There, but they're having us go out here. All right.
Is there a question? No. Okay.
And I go to Is this how we came in? I thought we came in from over there. I did too. Why is it telling me to go here?
Christine, please mute your microphone. I have to go.
Thanks. So what we have here on the left is the Near Creek zone, and on the right is the High Marsh zone. The black is the water level in the wells. So you can see that in the Near Creek, of course, there's a semi-diurnal tide. And the colors are the redox, the different depth redox sensors. So blue is the shallowest at 10 centimeters, and then it goes down to the deepest is yellow at 45 centimeters in the Near Creek, and then High Marsh only has 4 here, from 4 to 29 centimeters. And what you can see is that down to about 40 centimeters depth, we actually see these strong semi-diurnal variations in redox potential. Whereas in the High Marsh, it's much less variable, and at about 15 centimeters or so, it tends to be reducing. So a much thinner part of it is actually oxidizing. We think that this is actually the result of a crab burrow, a macropore that appeared and disappeared. It's not advancing again. There we go. And so in the different zones, we see very different redox behavior. And I didn't show you the interior, but it's because it's not interesting. Pretty much at all depths, it's reducing conditions except at the very, very top. And so, well, what does that mean if, oh, and I have the burrows on here, which the burrows play a role in this too, but we'll ignore that for now. But basically what that means is that if you have a shallower oxidized zone, then if you look at the carbon oxidation rate with depth, you're going to have a shallower zone in which you're going to be oxidizing the carbon, right? But if you have a deeper zone, then obviously you're going to have a larger volume of your marsh that has a high carbon oxidation rate. And so if we integrate under that curve for say 50 centimeters depth, then we get maybe 660 millimoles per meter cube per day, or meter squared per day, and about twice that if we have high carbon oxidation rates that reach deeper into the system. So each of these zones is not only different ecologically and hydrologically and in terms of their redox, but they're different in terms of their carbon sequestration potential, because if they're oxidizing the carbon, they're not sequestering it, right? So what that means, so that translates then into differences in both the horizontal and the vertical carbon fluxes. And again, I'm just going to focus on the vertical. And if we look at those carbon oxidation rates in the context of the measured redox, we can conclude that you have moderate to high carbon storage in the near channel zone, low carbon storage in the spring deep zone, and the highest carbon storage in the interior of the marsh. And so because we're able to make those physical or hydrological biogeochemical linkages, then we can use a physics-based model to try and predict how things will change into the future. And so this is a hydrogeosphere model. A hydrogeosphere is integrated surface water subsurface model. So on the left, I'm showing the grid and the colors represent topography. And so that's just the surface grid. And on the right, I'm showing the subsurface grid. And you can see the boundary here is that St. John's River. And here's the big tidal channel that goes through our site. And here's where we were making all the measurements. It's coupled surface, subsurface. Like I said, it's three-dimensional, variably saturated. And so we can, so we can force the model with tides, and we can also raise sea level rise gradually in the model. And so what I'm showing on the left is the boundary condition, the tidal boundary condition, and I'm showing on the right is the water depth. So of course the depth is deepest in the tidal channel, but you can see that periodically areas of marsh become inundated, and that happens on different frequencies. And so based on our measurements, we essentially, we can use fast Fourier transforms on the simulated hydraulic heads to characterize them and classify them as our different hydrological zones. So Julia did that, and she ran the model with different rates of relative sea level rise that incorporate both sea level rise and assumptions about the sedimentation rate or the compaction rate in the marsh. So this is a representation of the marsh zonation. So the blue is subtidal, and that's of course in the creeks. The light blue is tidal, that's the near creek zone. The green is the spring neap zone. The orange is the interior, and red is upland. And so this is what's happening now. So at zero relative sea level rise, these are the proportions that we have in this part of the model. And then as sea level rises, those proportions change. And so one thing to see is that the subtidal zone increases with sea level rise, and there seems to be a tipping point for the system at around 40 centimeters or so, where it really starts to drown a lot of the interior part of the marsh. And then the other thing to note is that the upland starts to shrink. And again, there seems to be kind of a tipping point around maybe 30 centimeters or so, where you really start to get marsh migration into the upland. And if we think about each of these zones having different burial rates, then we can make a connection to how the carbon sequestration potential of the whole marsh could change with sea level rise. And so that's what we did here, where we have the carbon accumulation rate with relative sea level rise. And the red is our present day, and here are the relative sea level rises. And so again, there's sort of this tipping point in terms of carbon accumulation around say 50 centimeters of sea level rise, relative sea level rise or so, where actually in the beginning with sea level rise, we increase the carbon sequestration potential, which makes sense, right? Because the water levels are going to rise, but at some point you're drowning marshes and losing marsh, and so that accumulation rate begins to decline. Another thing, so as a hydrologist, one thing I was really curious about is whether what's happening in the upland really matters to what's happening in the marsh. And so I think of coastal systems as being in one of two categories. One is topography limited systems, where the topography is low enough or the recharge rate is high enough that there's a pretty thin unsaturated zone. And so in a system like this, when sea level rises, the water table is already so near to land surface that the water table really can't rise in response. But in recharge limited systems, there's a thick enough unsaturated zone or a low enough recharge rate that if sea level rises, the groundwater table can keep up with it, right? And so it matters a lot for the subsurface seawater intrusion, but it also could matter for what's happening in the marshes. And so I typically think of Delaware as being topography limited. But in any case, we tested this idea by in one set of simulations holding the upland water levels constant, and in the other simulation allowing the upland to keep up with sea level rise, allowing 100% sea level rise. And so actually what I had showed you before was the 100% sea level rise scenario, but actually if it's topography limited, if the upland water level stays constant as sea level rises, the carbon accumulation rate is actually overall quite a bit less, but its variability is also less. So this tells us that you really have to think about the whole system in order to understand how the salt marshes are going to function with future change. And then the last piece of this work, see how I'm doing on time, is thinking about how the marsh migration feeds back into flooding and salinization. So I mentioned that one of the ecosystem services of marshes is that they protect the upland from flooding. And so what I'm showing here is actually one of our CZN sites. It's in Delaware. It's an agricultural site. This at the bottom is the salt marsh, and then here is our farm. The farmer actually didn't plant where we were working this year, so there's kind of this big bald spot, and that's our tent where we're working. But the point is that, so you see this healthy crop, and this is healthy marsh. Then there's Phragmites, which are invasive, and that's pretty much what populates the zone that's migrating. And then there's this zone here that looks kind of green, where it used to be farmed, but now it's too salty to farm, and so the farmer doesn't farm it, but instead he mows. And so you can look at it from above as well, if I can get this to advance. This is the same site where we're going from marsh through the transition to the healthy ag. And so here's the frag, and here's this zone where the farmer mows. And so the idea is, well, what if you allow the marsh to migrate, allow that frag to go inland instead of mowing? Does that actually protect your crops back here? And so Julia did some modeling similar to this. So this is another coupled hydrogeosphere model. I'm showing a storm surge in the surface water domain. I've decoupled them just so you can see. And the colors here are salinity. And so you can see that as the storm surge goes up and comes back, the salt water infiltrates and then flushes out. This is 20,000 days, so it actually takes years to flush out. I'll just show you that again quickly. At first, the time steps are small, so the salinization happens rapidly and the flushing out happens slowly. And so she used this kind of a model to then figure out or to calculate how much salinization is occurring behind marshes for different marsh widths. And she did this for 2 different storm surge heights, and she did it for 5 different land surface elevation gradients, as well as different total marsh widths. So marsh width is on the x-axis, and on the y-axis we have 3 metrics. One is the inland extent that the flood went to. One is the total volume of salt water that infiltrated into the aquifer. And the third is the volume of the aquifer that became salinized. And what you see in every case is that you get a decrease in these metrics as you increase the total marsh width. And the reason is that the marsh is it is essentially rougher than the crop, right? So there's more drag on the floodwaters that are coming from the marsh, which keeps them from going as far into the field, right? So this is essentially, in the model, changing a Manning coefficient. But so we get a decrease in the landward flood extent with an increase in marsh width and terrestrial slope. And of course, we get an increase in the flood extent with an increase in surge height. And the same is true for the salinization. And so we see that the marsh migration is protecting the farmland from surge flooding and saltwater infiltration and aquifer salinization, which is important because in a lot of these fields, you have irrigation wells, which are pumping. So if you salinize that soil, A, you're hurting the crops at the surface, but B, because it takes so long to flush out, you could salinize your irrigation water for years. And it protects the crop yields, right? So, and we know from the literature how the relative yield depends on the electrical conductivity or the salinity of the soil. And this is for corn and soybeans. And based on this and our model calculated salinities, we could infer the costs and benefits of allowing marshes to migrate. And so we see that the marsh migration can financially benefit the farms while also increasing ecosystem services. And if you value those ecosystem services, it's actually a huge amount of financial gain in the system as a whole. And so we think that this could have important policy implications with incentives potentially for farmers to allow marshes to migrate, allow their land that's no longer farmable to become marsh. So the insights that we got from this work is that if we can establish the links between the physical, chemical, and ecological ecosystem components, then we can use mechanistic models to forecast the future change. We see that the wetland zonation patterns are dynamically linked to relative sea level rise, as we would expect, but also to the upland groundwater table. So again, highlighting the importance of the regional hydrology and geology in the fate of coastal wetlands. And lastly, marsh migration into agricultural land could add protection in terms of salinization and provide societal benefits. So what are the implications of this marsh migration on nutrient fluxes and ecosystems? That's the topic of our relatively new project. Now we're actually in our 3rd year. This is a NSF-funded Coastal Critical Zone Network project. We call it the Coastal CZN. This is our team. We represent 6 different institutions, including BU, VIMS, University of Maryland Eastern Shore, George Washington, and the University of Maryland. So this is some of our group out in their natural habitat in the field. And one of the motivations for doing this work is that ghost forests are making the news pretty much everywhere along the Atlantic coastline. And so these ghost forests are basically stark images that show us actively that marshes are migrating, and marshes have migrated over time. So you can see these dead bleached trees that are coming out of the salt marsh. They used to be forests, and the salt marsh has moved in. And so this is really evident in many places, especially along the Atlantic coastline, mid-Atlantic coastline. And of course, I already mentioned the agricultural problems that saltwater intrusion can cause. And this is also something that's really making headlines. So the idea of the project is that these concurrent changes in water and chemical cycling are altering the function of the coastal critical zone. And so our cluster, this project is a cluster of sites, is quantifying these coupled processes and feedbacks that govern the hydrological, ecological, geomorphological, and biogeochemical transformations to understand how these shifts in the land-sea transition zone is going to translate to changes in the cycling and fluxes and storage of critical elements like nutrients and carbon at the land-sea margin. So what are the key drivers of these changes? Well, we've already talked a little bit about this, but we can think of it as one, slow hydrologic processes, so basically sea level rise, which can cause, of course, this response in the water table, which then can lead to groundwater flooding. It can also lead to long time scale seawater intrusion, and another slow process is that flushing process. And this is seawater intrusion from below rather than from above. And superimposed on that is fast hydrologic processes. So high tides, storm surge events that cause saltwater to move inland and cause infiltration of saltwater. So the idea in terms of what that does to the ecology is that if you have salinity on this y-axis and time on the x-axis, or you could think of it as distance from mean high water, if this is your salinity where your plants start to begin to be stressed, then if you have just fast processes, you go into that stress and out of it, into it, out of it. And so plants may be resilient enough that they're able to bounce back after those just episodic events. But at some point, if you superimpose slow processes, then you may hit a tipping point where the plants are spending so much time in and above that plant stress zone that they just die. So we're thinking about these fast processes and slow hydrologic processes. We're trying to link them to ecological and geomorphological response. And what do I mean by that? Well, if you have trees in this transition zone and they die, those trees have roots. And so the root zone may collapse, which leads to a collapse of the land surface elevation. So the land surface elevation will become lower, which of course is going to positively feed back with your flooding. And then we're also looking at biogeochemical responses of these processes. So if you have a storm surge, you're salinizing an area that was previously fresh, you can have ion exchange processes, you would have changes in redox, which would then, if you have iron dissolution, that can release carbon, it can release other sorbent constituents like phosphorus. So we have a network of sites. We have 3 study locations in the Delmarva. So the Virginia coast, which is exposed to the ocean, the Delaware Bay coast exposed to the Delaware Bay, and the Chesapeake coast. And at each location, we have 2 sites. One is in blue, a marsh forest transition, and in red, a marsh agriculture transition. And so I'm showing here the Virginia agricultural site. That's Matt Kerwin. This is an area of burnt out, salty soil. And this is our Delaware forest site where you can see we're in the marsh here, and then you can see the ghost forest. This is the Virginia Coast LTER. This is drone footage from Tyler Messerschmidt, who works with Matt Kerwin at VIMS. This is the LTER site, and then we're moving out toward the marsh. And so you can see there's healthy forest, and now it's transitioning into some dead trees, and then more and more dead trees proportionally as you get to the marsh, and then eventually the healthy marsh. So you can see that at this site, we have a really thick zone, transition zone, where there's active marsh migration into the forest. Oops. So I don't have time to go through all of it, but I just wanted to mention that we spent the first 2 years of this project instrumenting these sites. And we have hydrologic, ecologic, biogeochemical, and geomorphological instrumentation and measurements, and we're also going to plan some modeling. So I want to show you some preliminary data from our sites, and just give you a flavor of some of the measurements we're making. I'll try to do this quickly. So in each of these sites, we have 4 subsites from healthy marsh. So in ag, healthy marsh to healthy crop. We have, at each of those subsites, we have a well measuring conductivity, temperature, and depth. We have 2 soil moisture sensors. We have these multi-depth redox probes that I showed you before. We have pore water samplers and some other things as well. In the forested sites, we also have, so we have the same instrumentation, but we also have ecological measurements. So we have sap flux sensors that are measuring sap flux every 15 minutes, which is really cool. And we have these band dendrometers. This is Karen Gadan's work, where she's measuring the circumference of the trees as they have very small changes over time. We're also doing tree coring. And so this is just 2 of our sites to give you a sense of where we have the instrumentation. So here from healthy marsh through this transition zone, again, this burnt out zone, to marginal and healthy. It turns out, so when we first started doing, when we proposed the project, we actually proposed to have a space for time substitution, right? So what's transition now will be marsh in the future, et cetera. But it turns out that at this site in particular, these processes are happening so quickly that there's no longer healthy ag here. So after only 2 years, the farmer has decided it's not worth it to farm this property, and so it's no longer being farmed. And which at first we thought, oh no, this is going to be a problem for our measurements, but then we realized that this is a pretty good opportunity to really see in real time how these systems change. And this is the Maryland forest, again, from marsh up into the high forest. So in terms of hydrology, this is primarily my group and Sergio Fagarassi's group. We're looking at these slow and fast hydrologic processes, linking them to salinization and water table depths. So again, the wells with these CTD loggers and moisture sensors. Here's just an example of some of the data that we're getting. These are water table elevations in the Maryland forest. The orange and yellow are in the marsh, and then green's low forest, and the blue and purple are in the high forest. And this is depth to water. And this is the tidal stage in the red, and the blue bars are precipitation. So you can see that we've actually captured several storm events. So this was Hurricane Ida. This was a nor'easter and a big high tide. You can see the tide goes way up there. And then 2 nor'easters in a row here. And so we see the effects not only on the water levels, but also on the redox potential. And so you can see that during these storm events, we get some changes in the redox potential. Oh no, I'm sorry, this is salinity. So this is salinization. So the salinity goes up after some of these storm events, and then it actually stays elevated afterwards, and actually in some cases continues to rise. So recovery time is something that we're really interested in, because how long does it take this water to flush out? How long might the trees and the crops be in this zone of stress? So this is an example of differences in salinity recovery time. In the Virginia forest site, we have a lot of clay, really thick clay at the top, and this is representing the water table here. At the Delaware ag site, we have a little bit of clay, but mostly sand underneath it. And so at the Virginia site, we have the salinization event. This is in, I guess, October of 2021. The salinity stays elevated, for at least 6 months and longer. And in the Delaware ag site, you get a big increase too, but that comes down quite a bit in the next month or so, and then fully recovers. So the lithology, the geology of the systems is going to play a role in these processes. This is, again, Karen Gadan and Stephanie Stotts are doing ecological work. So they're looking at the relationships between tree water use and tree growth, and also the hydrologic conditions on short and long time scales. So on long time scales, dendrochronology, and on short time scales, these sensors. And an example of some of Karen's work, this is the top row here are different trees in the high forest, in the mid forest, and in the low forest. I think this is at the Maryland site. And you can see oh, and this is over time, right? So this is in December into February. So we're going into the winter here. So sap flow tends to slow for the winter. And there's overall lower sap flow in the low forest, although it does vary by individual tree, but much less sap flow or water going through the tree in the low forest than in the high forest. Matt Kerwin is leading up the geomorphology. So he's using sediment elevation tables and measuring really small millimeter scale changes in soil surface elevation and the thickness of the root zone. And we can tie that then to the ecological shifts. In biogeochemistry, we have several biogeochemists. So we're looking at again, the hydrologic biogeochemical linkages over space and time. And understanding the spatial and temporal chemical signature of the pore water and also the sediments. And Kate Tully and Yo Chin are focusing on the chemical signature of the fluxes out of the system with ISCO samplers in the tidal channels. So just some of the preliminary results. This is redox potential on the x-axis at these 6 different depths, so 5 centimeters down to 120 centimeters. The blue approximately represents the depth or the elevation of the water table. The blue here are the soil moisture probes. We only have them at 2 depths, and they have corresponding redox sensors. So the white is the EH. So you can see that EH is fairly elevated at the surface, as we would expect. And then a little bit lower once you get deeper into the subsurface. But the EH really responds to the amount of moisture in the system. And so we started a very dry period starting maybe here or so, where you can see here that we have increases in the soil moisture, right, probably from precipitation events, but then it really starts to decline. So there's this long period of dropping soil moisture levels. And at some point that triggers an increase in the redox potential at different depths. And it varies by depth, a little bit delayed as you go deeper in the system, but there is a response to the soil moisture. There is also a difference in redox potential with the lithology. So similar to the response of the salinity, the lithology also plays a major role here. So again, in the Virginia site where we have this thick superficial clay, the orange and the yellow are the redox probes that are above the water table approximately, and then you go deeper into the clay below the water table. So below the water table, it's generally reducing conditions, and the shallowest one hops up periodically. And once we get into, again, that very dry period, then the 20 centimeter one starts to come up, and then the 30 centimeters. But we can contrast that with the Delaware Ag site, where again, we just have a little bit of silt and clay at the top and then sand below that. The water table here is below approximately this yellow sensor. And you can see that there's a huge amount of variability, especially in the shallow sensors compared to the Virginia site. But also the really interesting thing is that the deepest one actually has the highest redox potential. And we think that's because it's in the sand. And so you're getting preferential groundwater flow through that sandy zone that's bringing in probably more oxic water. Whereas in the clay, you have these organics, you're going to have reducing conditions, and you have much less flushing. I am about at time, so I'll try to be really quick here, but this is some of Sean Fetro and Angelia Seiferth's work, where they're looking at the signature of the pore water at these different zones. And so they see a clear salinity gradient from the marsh to the high forest, and also some variations in depth. This is redox, so we're measuring it in the probes, but we're also measuring it in the pore water. And so again, he sees an increase in redox from marsh to the high forest, as we expect, but also some really interesting changes in the redox. So similarly to what I just showed you with the probes, where we see higher redox or EH at depth in that sandy zone, he's also seeing that in pore water. The solute flux measurements, we're using ISCO samplers to measure water flux, as well as dissolved organic and inorganic carbon, nitrogen, phosphorus, chloride, sulfate. And we're also using fluorescence and absorbance spectroscopy to look at the origin of the carbon and the quality of the carbon. This is led by Kate Tully and Yo Chin's groups. And so just to give you a sense of, so the ISCOs we can trigger from our cell phones, and we usually have them programmed to take a sample every hour for a 24-hour cycle. So that would be either 2 semi-diurnal tides or a storm. So we've been triggering them to both to see typical tides as well as high tides and storms. And so this is actually 24 hours during Hurricane Ida. High tide occurred approximately here. I'm showing dissolved organic carbon concentration versus hour. And you can see at the high tide, we have the lowest concentration, so generally an inverse relationship between DOC and the tide. And so there are a lot of reasons that could happen, both hydrological and biogeochemical, and that's what we're trying to understand. Similarly, if we look at the UV absorbance, which tells us approximately the origin, whether it's more terrestrial or more marine, of the dissolved organic carbon, you can see that that also changes similarly with the tide. And we have some sites, they have similar patterns, and then we have some that are a bit mysterious. So again, this is preliminary data, but we're hoping to be able to untangle some of these questions. So work is ongoing to link the hydrologic and the redox shifts to those changes. And then lastly, I'll say, you saw this already, but we're using the same kind of modeling. The idea is to, in the out years, once we get data collection, to do some modeling where we're coupling these mechanistic models of flooding and salinization to models of marsh geomorphology to try and get at some of these mechanistic feedbacks between the vegetation, the hydrology, and the geomorphology. And so we're trying to put these processes together. I didn't talk about it, but there's also, a human component here. And we really need to consider these things together to predict the changes in the fluxes at the land sea margin. I just want to mention briefly, if anyone is interested in working with us, we have some money to support travel, although you may not be traveling far, but travel to work with us at our sites. Feel free to visit our website to learn more about that, or just email me. We also have undergraduate internships starting this coming summer. So we have 6 available at all of our research sites and institutions. And please, again, contact us if you're interested. And so I'll just close to say, what I've been saying before, which is basically we have to think about the different system components. We have to think across scales, across different settings, to really understand, and also consider what humans are doing to really understand how our coastlines are going to change and what that means for fluxes of carbon and nutrients, et cetera. So thanks.
Thanks so much, Holly. That was really fascinating. I had a chance to go on a field trip last August and learned a lot of things, but I learned a lot more things today that went well beyond the things you talked about there. So it's really fascinating study. I'm hoping we get some people to raise questions. So we're opening it up now for question and answer. And since I can't see everybody, the best thing would be if you just post in the chat that you'd like to ask a question. And if you then want to unmute and ask your question directly, we'll call on you in the order in which these things show up. Since nothing's shown up yet, people are just starting to think about this. I'm going to start by kind of backing out a bit to the big picture. So, it was a big picture already, right? So this is a huge landscape change that's affecting, obviously, global systems as well as systems in this area, but there's particular interest in, certainly here in the Chesapeake Bay watershed, about the whole restoration effort that's going on. How are we doing in meeting our targets? And so there's a couple of pieces of what you talked about that I think, and Kathy Boomer is going to ask a question. She'll be up, I guess there's 2 people in here. So one is just how these changes in biogeochemical fluxes actually, you think over a longer term might actually be affecting carbon sequestration, anything else to do with how nutrient flux behaves. The other question has to do with thinking about, I'll just leave it at that one right now because people want to ask other questions.
Yeah, so your question is, how are they changing, or
Yeah, if we project these things forward, right? You're modeling what's happening right now, but if we project forward, several decades, this whole thing looks like it's going to accelerate. And so, how do we view what this landscape's going to look like and what's going to be happening to these processes on a large scale, at the scale of this whole area?
Yeah, well, that's a good question. That's a hard question, right? That's basically why we're doing this study. Is I think that we first have to understand the mechanisms of what's happening. We have to understand how these processes are affecting the carbon and the nutrients and other contaminants before we can start to scale up in space and scale out in time. And so as a modeler, I keep thinking, well, if we can get at these mechanisms, if we can really, as a hydrologist, a physics person, I think it's all tied to the hydrology, right? That's my bias. But if we can link those processes that matter for the fluxes, right, for the biogeochemistry, for the ecology, if we can link those to something that we can model, in my view, more easily, which is the physics, the physical flooding and salinization processes, then we may be able to do a better job of scaling up in space and time. And so, I see that these site-specific projects, site-specific new understanding that we get will help us to gain the insights that we need to make those connections to be able to scale up. And by doing it not just at one site, but at 6 sites and, replicates of three of each different kind of site, even though they do have some differences in terms of how vulnerable they are to the storm surges based on where they're situated. They have different slopes, et cetera. So they have some differences where we can get a sense of variability, but they're also all on the Delmarva. And so it's a similar setting where we can hopefully draw some understanding we can then really scale up through models.
Okay, thanks. I'm going to go to the questions. The first one is from Julie Reichert, Nguyen, and then we'll have Kathy Boomer after that.
Thanks. Yeah. Thanks, Holly. Great presentation and much needed research to understand these mechanisms. I'm with the NOAA Chesapeake Bay office and I coordinate the climate resiliency work group with the Chesapeake Bay program. And there's been a lot of conversations around blue carbon marketing and can we utilize sort of that potential funding avenue for supporting tidal wetland or marsh restoration type projects. But what we've encountered is this dealing with the complexity of these fluxes, if we really need to have a better understanding. So I think your research is definitely helping and your team is helping with that. But I was just wondering if you had any insights on that, because since the marshes are vulnerable to sea level rise, and in order to enter the carbon market, you have to have some sort of confidence that you're going to sequester that carbon. Is there a certain site condition that it sounds like maybe clay, perhaps there was less carbon flux? I don't know. I was trying to follow the results of the presentation, but I don't know if there's any insights right now of sort of longevity of carbon sequestration, given all this variation that can happen with these biogeochemical processes.
Yeah. I mean, I think it's really hard. To some extent, we don't even know if some marshes are net loss of carbon or net gain of carbon. It's a really hard and complicated problem. But I mean, if we're able to at least show that some, that certain kinds of wetlands, as you say, are sequestering a certain amount, then of course, there's going to be variability, but at least maybe we can have that as a basis for valuing the wetlands in terms of carbon. I mean, I'm thinking the analogy of the Chesapeake Bay program in terms of nutrients, right? There's this Chesapeake Bay model, it's a parameterized model where you're estimating the nutrient loss in various places, but and of course it's not going to be perfectly right, but it's based on a lot of science that goes into it that allows these estimates. And so, I think we can hopefully get to the point where we can do the same thing in salt marshes, that we can at least understand what the major factors are that govern the carbon dynamics and use that to maybe develop smart policies.
Okay, thanks. Thanks very much. Kathy Boomer's up next. Kathy?
Holly, great presentation. It's so cool to see how you're using diverse data to evaluate nested systems and the interaction of those systems and in terms of driving ecosystem services. My question builds on Andy's and just to probe, how do these conceptual models apply to more terrestrial freshwater systems?
Yeah,
Do they apply?
Kathy. I mean, it's analogous, right? In terms of how hydrology and biogeochemistry and ecology interact. It would just be a different system. So in the coastal systems, we have salinity as a factor. So that's something that wouldn't be as much of a factor in upland systems, with the exception maybe of urban systems where you have a lot of salinity coming in with stormwater and things like that, or road salt. Which is, I think, an interesting, actually, link maybe between the two CZN projects, the urban and the coastal CZN projects. But yeah, in any wetland, as you have water level changes, they may not be tidal, but they may be on different timescales. You're going to have changes in biogeochemistry, and understanding how those link together is really important for predicting how they'll evolve in the future, whether it's due to climate change or due to human impacts, etc. I don't know if that answered your question.
No, I did. I think we often don't think about both water quantity and water quality affecting carbon sequestration and nutrient dynamics. So this is, I think, a really neat example that we might apply and to use for studying wetlands and better understanding where they can provide the benefits that we are seeking.
Thanks.
Okay, thank you. Does anybody else have a question they would like to ask? So I'm just going to run one more by you because we still have another minute or two. One of the most interesting things I saw on the field trip was this example that you alluded to it only by showing a picture of the installation. The sap flow work that's being done in some of these trees that are just inland from the marsh. One of the most fascinating things I learned was when they described to us what happened with coastal inundation event that actually caused a reversal of the osmotic gradient so that sap was flowing down out of the tree and causing stress on the trees. And that kind of effect and its influence on how rapidly the forest might die out and what the implications are for the rate of retreat of the forest, to me was a really interesting phenomenon. I wonder, since most people here weren't on that trip, you could just say a couple of words about that particular topic and what its implications are.
Yeah, so maybe I should have taken Karen's slides and put those ones in instead of the other ones. But yeah, so right, there is this, I mean, the sap flux sensors, as a hydrologist and not an ecologist, I think they're so incredibly fascinating. And they tell us so much. But so, right, so like Andy said, we had a storm surge event. It flooded and salinized the trees that were instrumented with the sap flux sensors. And what Karen measured was that instead of, typically if you have a lot of water in the system, maybe you would expect that you would have more evapotranspiration, right? Which would mean more, the sap flux is basically the water moving up the tree, right? But in this case, we actually saw that the flux of the sap was downward. So the roots were like leaking water, or the soil, maybe the soil was pulling it out of the roots. And as far as I know, we don't exactly, and again, I'm not an ecologist, but as far as I know, we we don't exactly know why this happened, whether it's some sort of osmotic force because the soil water became more saline, or whether it's a different physiological response from the tree. But it is a major problem for the health of the tree, because if you get that reverse flow, you can actually get cavitation within the tree, right? So you get really low pressures in the tree that can actually harm the tree itself. And so if that happens to an extent, you know, to some threshold, then the tree will die. And so again, there's sort of this fast process and then the slow process of change that is conspiring to affect the health of the trees. And I think this is the kind of information that can really help us understand it. And from a hydrological perspective, there's a feedback too. So understanding how much sap is going up the trees, that's a measure essentially of, or a proxy for evapotranspiration or transpiration. That feeds back into the hydrological system, so, which also feeds back into the salinity. So if you have a lot of transpiration, you may be having more salinity in your soils. So so again, I think, you know, looking at how this whole system is functioning is really giving us new insights.
Okay, thanks. We are at 102. I'm not seeing any more questions in the chat, so I think I will just let people know we are recording this seminar. Once we stop the recording, it will come back with a link, and then we will have it posted on the YouTube channel of UMBC's Center for Social Science Scholarship. So I will send out a link to that, because I know there's a number of people who said they were interested in trying to attend but couldn't because of schedule conflict. So Holly, thanks so much for a really fascinating presentation. And I hope more people see this and learn more also about what's going on with the entire coastal critical zone network, and maybe you'll get some people contacting you to ask more questions about it. So thanks
Sounds
very
great.
much.
Thanks so much, Andy. Thanks, everyone.
Okay, I'm going to stop the recording now, and we are stopping now.